students, we used to always make nothing but hundreds of my cell biology exams. I couldn't quite, told him I was going to bring him down. I couldn't bring him down. Okay? I said I was going to bring him down and he would make 102. Okay? So he's a senior who's graduating with the 4.0 average. Okay? So he gave a wonderful seminar last Friday. So we have two guys who are giving seminars today at 2 o'clock. So come in and see what you're going to have to do when you get to be seniors. Okay? Come in for one of them. All right, so you got your quiz. Yes, sir. Okay. So I'm going to, did you have a homework assignment for today? If you don't have it, that's okay. So I'm going to talk about two things today. I'm going to talk about how we make a protein in the cytosol. How we make proteins in 3 polycycles. This is sort of a general view. So I'm not going to talk about tRNA. Now, if I gave you a detailed view of this, I'm not going to talk about the role of tRNA. I'm not going to talk about the role of tRNA. Okay? So this is a general view of how we make proteins on 3 polysomes in the cytosol. And then I'm going to talk a little bit about how you transport some of those proteins into the mitochondria. Okay? So that's the theme of today's talk. So let's begin. And so most of you should know this story. Perhaps not quite the way I tell it. All right, so let's begin with a messenger RNA. Let's begin with the messenger RNA. I'm going to try to do this large enough so it's clear to everybody. So here's a messenger RNA molecule that's floating around in the cytosol, waiting to be translated. Okay, it's got a 5' end, it's got a 3' end. So it has a cap structure at the 5' end, it's got poly A at the 3' end. Now, we're going to come back and talk more about those ends in detail later. Okay? So here's the messenger RNA, here's the 5' end, and we're going to translate this from its 5' end to its 3' end. Okay? So it has a cap structure at its 5' end, at its 3' end. We have poly A. So again, just to be clear, poly A is a sequence of 100 to 200 adenylic acid residues, adenine. Okay, so let's go ahead and write this down. Okay, we'll describe cap structure later. So this is 100 to 200 adenylic acid residues, adenine acid residues. And so all messenger RNAs have this structure. This is the only RNA that has this structure. This is the only RNA that has the cap structure. So it must be fairly important for messenger RNAs. Okay, so that's the message that's floating around the cytosol. This is what we call mRNA. Again, all of this should be really old hat to you. All right, so what we're going to do is translate. So the messenger RNA has been transported into the cytosol through nucleopores. The subunits have been transported into the cytosol through nucleopores. And so they're going to meet up in the cytosol, and we're going to engage in translation. We do transcription in the nucleus, we do translation in the cytosol. Okay? And so the subunit that starts the process is the small ribosomal subunit. So an intact ribosome does not bind to the 5' end. The 5' the small subunit binds initially. So here's a small subunit floating around in the pool. Here's the 40S subunit. Okay? And here's the large ribosomal subunit floating around in the pool. So this is 60S. Now, it turns out that the 60S has a groove in it that the nascent polypeptide chain grooves in. Okay? So by nascent, we mean newly synthesized. Okay? So this is a groove. So as the polypeptide chain is going to be made, or is being made, it's in this groove. Okay? So this is a groove. And let's just say which holds 30 to 40 amino acids in the nascent polypeptide chain. The nascent Nascent means newly synthesized, nascent polypeptide chain. And all I'm saying is that this length of the groove is sufficient to hold about 40 amino acids in the polypeptide chain. Okay? All right. So the 40S subunit is going to bind to the 5' end. And one of the functions of the cap subunit is that it serves as part of the binding site for the small subunit. So the small subunit now binds to a site that includes the cap structure. It's one of the functions of the cap structure. It's a part of the binding site for the small ribosomal subunit. So 40S is now on. Okay? So here's 40S. Okay? And so 40S is now going to move along the message. So the 40S now moves along the message. And so here it is at a certain point in the message. And that point that it stops briefly has a codon that says AUG. There's a codon that says AUG. So we're going to stop briefly at a codon that says AUG. Now this is the start codon for protein synthesis, AUG. So it not only tells the whole polysome that we're going to start making a protein here, but it also specifies the first amino acid in the polypeptide chain. So two functions of the start codon. So this now we call the start codon. And we're saying that the start codon has two functions. So two functions of the start codon. So one is that it says begin protein synthesis. And I'm going to write this differently. Begin translation. Begin translation. And the second is that it specifies the first amino acid in the polypeptide chain. Specifies the first amino acid in the polypeptide chain. So specifies the first AA amino acid in the polypeptide chain. Two important functions of the start codon. Okay, so when the small subunit reaches the start codon, the large subunit binds to the start codon as well. And now, for the first time, we have an intact ribosome. We didn't have an intact ribosome before now. Okay? So now one of the questions that I always trick students, or trip them up on, is that I say that an intact ribosome binds to the 5' end of the message to begin translation. Is that true? An intact ribosome binds to the 5' end of the messenger RNA to begin translation. Is that true? That's not true. Okay, so if I ask, put that on an exam, you say no. Okay, it's the small subunit, and so we only make the intact ribosome when we get to AUG. So here's the so the small subunit went on first, and now the large subunit is going to go on. And so now we have the large subunit on, and here's the groove in the large ribosomal subunit. And so AUG now is going to code for the first amino acid in the polypeptide chain, and that's always methionine. Methionine. Okay? So here's now the first amino acid that we put into this polypeptide chain, and an amino group, and I'm going to draw this in a moment. An amino group defines the N-terminus. Okay? So this now is the N-terminus of the polypeptide chain. Okay? And so this particular amino acid here is called methionine. Methionine. Now let me just do a slight diversion and talk a little bit about the amino acid structure. Okay? So let's I want to show you the structure of methionine. Okay, but let's start by talking about the general structure of an amino acid. So the general structure of an amino acid. So general structure of an amino acid, an AA. All right, so this is something that all of you know. So we have an NH2 group, we have a C, we have a carboxyl group, we have an H, And then we have an R group. It's a general structure of an amino acid. So we call this particular amino group, which is attached to this carbon. Let's go ahead and define this carbon first. This carbon is called the alpha carbon. And so if you're attached to this particular carbon, you're going to be called the alpha somebody. So this amino group here is called the alpha amino group. This group is called the alpha carboxyl group. So this now, this is the alpha amino group. This is the alpha carboxyl group. And the group that makes the different amino acids unique is this R group. And so it turns out that there are 20 different amino acids that are encoded in the genetic code. 20 different amino acids that have different R groups that are encoded in the genetic code. Okay? So 20 different AAs are encoded in the genetic code. Encoded in the genetic code. Now what I want to do is look at one of them. I want to look at the structure of methionine. Anybody have any questions about what I've said? So this is the general structure of all amino acids. Okay, except maybe one. There's one that's called proline that looks a little different. So now the structure, let's write it here for the moment. Structure of methionine. So this is methionine. I'm going to have to erase this in a minute. So this is the amino acid now that we start all protein synthesis in the cytosol. Okay, methionine. So this is what it looks like. So NH2, CH, COOH, CH, H, CH, H, S, and now CH3. CH3. Okay? So this is the R group of this particular amino acid. This is the alpha amino group, and this defines the N-terminus of the polypeptide chain. This amino group is going to be at the N-terminus. So when I talk about this N-terminus, I'm talking about this particular amino group. So this defines, or this amino group is at the N-terminus, of the polypeptide. Again, all proteins that we make in the cytosol begin with methionine. Now the interesting thing is that proteins that are made in mitochondria and bacteria begin with a variation of this particular amino acid that's called N-formyl methionine. Okay, that's sort of interesting. So let's draw that structure. Here's N-formyl methionine. So it's a very simple variation on this. It's NH, and it's got a formyl group attached to this N. This is N-formyl. Now we have CH, COOH. And so I'm going to just sort of simplify this and say CH2 taken 2 times. Okay, I hope you see what I'm doing. And now S, now CH3. This is N-formyl methionine. Interesting thing is that protein synthesis in bacteria and in mitochondria begins with N-formyl methionine. Protein synthesis in the cytosol begins with methionine. What's that sort of suggesting you?
That bacteria probably use N-formyl methionine, or
So I can't hear you.
that bacterium probably use N-formyl methionine, or that there's a relationship with that.
What does related to what we've been talking about? What is this? And let me ask you this: what is this supportive of? Related to what we've been doing.
Endosymbiosis.
It's supportive of endosymbiosis. How is that the case? How is it supported by endosymbiosis? Somebody's got to help me here today because of my energy is flagging. How is this supported with endosymbiosis? We keep talking about different pieces of evidence that support endosymbiosis. How would this support endosymbiosis?
Bacteria did the same thing.
I can't hear you.
If other large prokaryotes did the same, use the same.
So mitochondria are behaving just like the cells that we think that they originated from, right? That's the point. Okay, we keep making that point. Here's another piece of evidence that supports the endosymbiont hypothesis. All right, let's go ahead and write this down. So we say the informal methionine is what you start proteins with in bacteria. So we say in bacteria and mitochondria. And this supports the endosymbiont hypothesis. Additional evidence that supports the endosymbiont hypothesis, okay? Supports, so I'm going to erase all of this in a minute, supports endosymbiont hypothesis. So the hypothesis now is well established, and we don't call it a hypothesis anymore. We call it a theory, okay? Endosymbiont theory. Okay, anybody have any questions about what I'm saying? Okay, so the ribosome, we now have an intact ribosome. So the ribosome is going to start reading the information in these codons. And as it reads the information in these codons, it's going to put more and more amino acids into the nascent polypeptide chain. Okay, so let's move this ribosome along. It's going to read the codons, and each one of those codons is going to specify an amino acid. Okay, so we're going to move along. And so we've read this much of the polypeptide chain now. And so the polypeptide chain now has grown. So here's the polypeptide chain. So it's now filling up the entire groove. Here's the N-terminus. And now we've made all of these amino acids. We've read this information and converted that information into these amino acids. Okay? Nascent polypeptide chain is still in the groove. Okay? So let's call this now, let's call this a nascent polypeptide chain. Nascent polypeptide chain. What do I mean by nascent? Say it a couple of times. What do I mean by nascent? Say it loud. Newly synthesized. Nascent. Newly synthesized. Okay? All right. Yes, sir.
So I understand this whole thing, okay, about how one ribosomal subunit can have a thymine. Fine. But when there's other codons along the mRNA, how does the ribosomal subunits know, okay, I'm going to go here, or you're going to go there?
So it's going to be going right along. It has to go in this direction.
So as it goes along, does it just have
It's got tRNAs that are going to bind to the subunit and are responsible. Each tRNA is going to have an amino acid associated with it. And it's the tRNA that's going to recognize the codon.
Okay.
All right?
So this mRNA is between these two subunits, and the tRNA is what's going to...
So the tRNA is going to sit, and we'll come back and talk about this perhaps in detail. We did this in general biology. tRNAs are going to sit on the ribosome and read the information in the codon.
Okay.
And so the ribosome has the machinery now to take the amino acids off the tRNA and tie them together in a polypeptide.
Okay.
You with me?
Okay.
Other questions about this? All right, let's read a little bit more of this messenger RNA. So let's move further. So we read the messenger RNA from the 5' end to the 3' end. This is also the direction that this messenger RNA gets synthesized in, from 5' to 3'. Okay? So we're reading from this end to this end. So we move a little further. So here we are, having read more codons. And now I'm going to have to start erasing this. And so now this nascent polypeptide chain projects beyond the groove. So here's the nascent polypeptide chain. And so it now begins to do what? it now begins to do what? Fold. So at the same time we're synthesizing the nascent polypeptide chain, it starts to fold. And we say that anything that happens during translation is co-translation. So folding of the polypeptide chain is co-translation, okay? So this now is folding. Folding is co-translation. Anything that happens at the time that we make the protein, we say it is co-translation. Now it doesn't fold by itself. Although if we put this protein in the test tube, it has all of the information to fold into the correct structure in cells and folds with the assistance of other proteins that we call, anybody know the name of these proteins? Chaperones. Chaperones. So that was a discovery that we made, I guess, 20, 30 years ago. It's sort of major because we thought until then that proteins folded in the cell by themselves because they have all the information to specify their final structure. It's in the amino acid sequence. But in fact, they need proteins to assist them in the cells. We'll talk about why that's the case later. So here's one of these proteins. And so this is the major one that's involved in folding during translation. So this is a chaperone. So this is a member of the class of chaperones. And this particular protein goes by the name of HSP70. HSP70. So this protein is HSP70. HSP stands for heat shock proteins. 70 stands for the molecular weight of this protein in kilodaltons. Okay? That's right. So HSP, and this is the first class of chaperones that we discovered. These are heat shock proteins. And anytime you see a number in the name of a protein, it is the molecular weight of the protein in kilodaltons. So 70 now is equal to the MW in kilodaltons, where a dalton is the weight of a hydrogen atom. So this would be, in daltons, this would be 70,000 daltons. This is the weight of this particular protein. Anybody have any questions about what I just said? So HSP70 is an important chaperone that binds to proteins during the time that they are being synthesized and helps to fold. All right, let's just skip all the way down and sort of terminate this process. Skip all the way down to the end. So we've been folding all along the way, and so we get to the end. So how do we know where to stop? How do we know where to stop?
Stop codon.
Did I hear something?
Specific stop codon.
Stop codon. And so, who are the stop codons? I figured that this is old hat. Who are the stop codons?
UAG.
UAG is one of them. Let's explain what it is. So we get to the end of the, and so this is not, so there's always some additional nucleotides beyond the stop codon that we don't translate. It usually has some important information out there, but it's not part of the protein that we're making. So UAG is one of the stop codons. Turns out that there are three. And so I always think of the University of Georgia as one possibility. So this is UGA, and then the other is UAA. Okay? So those are the three stop codons. So when a ribosome sees any one of those, it's going to end translation. Okay? So we reach this point now. Here we are at this point. Here's a small subunit. Here's a large subunit. Here's the groove and the large ribosomal subunit. And so everything is going to fall apart at this point. Everything is going to fall apart. Let's just see what the products are. Let's erase this stuff. So everything comes apart at this point. So the nascent polypeptide chain is released, and so here it is now completely folded. So here's the C-terminus, which is the last part. And the C-terminus is the carboxyl group of the last amino acid in the polypeptide chain. It's this alpha-carboxyl group. This is the C-terminus, the last amino acid in the polypeptide chain. Okay? So the C-terminus, the protein, we're talking about most proteins here. Most proteins now, but not all, are completely folded. So this is in its tertiary structure. Okay? And so let's dissociate the chaperones. So they finished their job, and so here's HSP 70 coming off. And the ribosomal subunits now dissociate and begin to float around again in the pool of ribosomal subunits looking for the 5' end of another message. Okay? So this small subunit comes off. Here it is. Now back in the ribosomal subunit pool. And here's the large subunit having dissociated back in the ribosomal subunit pool. Okay? Now, is there a question about this? So this is the way we make the typical protein on a free polysome of the cytosol. Is there a question about this? Now, let's just talk about let's talk about mitochondrial proteins. So it turns out that mitochondrial proteins do not fold. So they don't fold because certain proteins associated with them that prevent them from folding. So let's put one of those up here. So a mitochondrial protein will not fold. And it's important that it not fold. So this is true for both proteins, but here now we have a mitochondrial protein. So here's a mitochondrial protein. So it's still basically in a denatured form. Here's its C-terminus, here's its N-terminus. Okay? Now it turns out that these proteins actually are pre-proteins. So mitochondrial proteins are precursors to the final proteins that we make in mitochondria. Okay? So this is a mitochondrial it's a precursor, pre-protein. So we make all mitochondrial proteins as pre-proteins, and when we get them into the mitochondria, we cut a piece out of them, and one of the pieces becomes the functional protein. Okay? So now this protein, unlike these, did not fold because it has proteins associated with it. Okay? So let's say prospective mitochondrial proteins do not fold during translation. Do not fold during translation. And the reason for that, again, is that they have some proteins associated with them that prevent them from folding. Now, the thing that's really puzzling to me is that one of these proteins is HSP70. One of these proteins is HSP70. So here's HSP70. So HSP, and this is true of all chaperones, they seem to have dual roles. Some cases, they help proteins fold. Here's HSP70 helping this protein fold. In other cases, it's preventing protein from folding. All right? We have to come back and try to see if we can figure that out. Okay? So now, for some particular reason, mitochondrial proteins are not allowed to fold. We keep them basically in a denatured state before we get them to the mitochondria. Now, the question that really is interesting to me is how do we know where this protein is going? How do we know that this protein is directed to the mitochondria? How do we ensure that this protein gets into the mitochondria? Yeah.
How do we ensure that it gets into the mitochondria?
Yeah. How do we? So of all of the organelles floating around, how do we direct this protein to one in particular?
Well, if it has the informal methionine, it could be like
It's not that.
Signals?
I can't hear you.
Signals?
Say it loud.
Signals.
Signal. Somebody else pick up on that. It says signal. What does that mean? I'm going to try this again and see if I don't fall.
it'll tag the protein.
So this protein has to have a signal on it to get it into a particular place in the cell. And so what must be characteristic of that place in the cell?
Let's have a receptor.
It's got to have a receptor. And so the work of Gunther Blobel actually led us to this sort of wonderful idea that all proteins have to have a signal in them so that they go to the right place. Now, let me just tell you how complicated this is. So free polysomes in the cytosol are making proteins that have a number of destinations. Let's list some of the destinations of proteins made on free polysomes. Okay, and this indicates the complication here. So proteins made on free polysomes, where do they go? So here we are. Destinations of proteins made on free polysomes. Made on free polysomes. So one is the cytosol itself, the cytosol. Okay? So one is the cytosol. And so an example of proteins that are made on free polysomes that are in the cytosol is the glycolytic enzymes. Where did we say glycolysis occurs? Where does glycolysis occur in a eukaryotic cell?
Cytosol.
Cytosol.
Right.
So the proteins, the enzymes that catalyze the reactions in glycolysis are made on free polysomes, and they stay in the cytosol. Okay? All right, so example, for example, glycolytic enzymes. So another destination is nucleoplasm. So all of the proteins basically inside the nucleus are made on free polysomes. Okay? So a second destination is nucleoplasm. And so give me an example of some proteins that we're talking about here. So give me a couple of examples of proteins in the nucleus.
Telomerase.
Telomerase, right? What else? What else have we? What are the major proteins that we've been talking about in the nucleus?
Lamina.
Ribosome.
Lamina.
So ribosomal.
RNA.
No,
Oh no. Ribosomal subunits.
no, sir. Protein. Ribosomal proteins. What else? What's one we talked about that's associated with chromatin? Say it loud. Histones. And is there another class of proteins associated with chromatin?
Non-histone.
Non-histone chromosomal proteins. Where are those proteins made?
Nucleus.
The nucleolus. Did you say that?
Nucleus.
Where are those proteins made?
The nucleus.
They're made on polysomes in the cytosol.
Oh,
How do we get them into the
the
nucleus?
nuclear pores.
Nuclear pores, right? Let's put this stuff together. Okay, so let's put a couple of examples here. Nucleoplasm, histone, ribosomal protein. Okay? For example, histone, ribosomal protein. Everything, all proteins in the nucleus. So we'll talk about more. So, third, which is what we're talking about now, is mitochondria. So all proteins in the mitochondrium, are made on free polysomes. A fourth is chloroplast. Fourth is chloroplast. The chloroplast. And the fifth is these organelles that are called peroxisomes. So, the question is, how do we target some proteins to the nucleoplasm? How do we target other proteins to the mitochondria? Other proteins to the chloroplast? Other proteins to the peroxisome? How do we do that? Somebody just sort of wrap this up for me, because I'm sensing that I'm not getting through here. I'm sensing that I'm not feeling. So how do we target specifically proteins? They're all made on the same polysomes out here in the cytosol, but yet they're different destinations. How are we going to do that? We just talked about
Different tags for different receptors.
We're going to do what?
Use different tags that
We're going
correspond
to
to
put
different
different tags or different. What's the other word we use?
receptors.
Other word we've used
Signal.
instead of tag,
Signal.
signal. We're going to put different signals in the proteins, and they're going to be different receptors at their destination. Okay? All right? So let's indicate that this involves, all right, so specification. Let's write it like this. Specification of the destination of a protein depends on signals and receptors. Depends on signals and receptors. Now I'm going to show you that this is even more complicated. So let's say we want to get a protein into the mitochondria. Let's look at the mitochondria for a moment. Look at the mitochondria for a moment. I'm going to erase this now. So we want to get a protein into the mitochondria. And so this is a very complex organelle. So here we go. So here's the outer membrane. So outer membrane of mitochondria. Here now is the inner membrane. Okay, so the point is that some proteins are transmembrane proteins in the outer membrane. So here's a protein, and all copies of this protein are in the outer membrane. All copies of this protein are in the outer membrane. Okay? Here's a protein that's in the inner membrane. All copies of this protein are in the inner membrane. Here's a protein now that's in the matrix. All copies of that protein are in the matrix. Now, let me just draw this a little bit differently. So let me just draw it as a folded protein. So here's a folded protein. Here's the N terminus. Here's the C terminus. All copies of this protein are in the matrix. Okay? Show another one. Show one more. So here's a protein that's in the inner membrane space. And all copies of this protein are in the inner membrane space. So this indicates the complexity of our problem. And so what do I mean by saying that this indicates that the problem is even more complex than what I've illustrated over there when we talk about the mitochondria?
In inner membrane space, how do you get to the
So
matrix?
we have to put a tag, we have to specify that this protein goes here. So it's not just sufficient for us to say mitochondria. We've got to say mitochondria and go to the outer membrane. Mitochondria and go to the inner membrane. Mitochondria and go to the matrix. Mitochondria and go to the inner membrane space. You with me on this? Mm-hmm. So this means that these proteins from the mitochondria probably have what? I can't hear you. They probably have what? Just based on what I just said. They probably have more than one signal. Are you with me on that? Proteins probably have more than one signal. Okay? So the typical mitochondrial protein, the typical mitochondrial protein has more than one signal. All right, so unfortunately I have to stop here. But we're going to talk about how one protein gets into the mitochondria when you come back. Now, yeah, when you come back here, there may be a pop quiz waiting for you. Okay? Because I like this. I like this. And you know, you're getting all these points at my expense. You get 4 out of 5, 5 out of 5. Keep doing it. Anybody have any final questions about this? I think this is kind of deep and interesting. And I'm sorry that you don't. Okay? But we'll get there. Okay, so enjoy spring break. Hope the beaches in Miami are warm and so on.